Hi, everyone. Today's lesson is on the cell cycle. So the cell cycle is a process by which cells reproduce. We've talked about this before. Essentially, we have a parent cell splitting into two daughter cells. Remember that for all regular body cells, the daughter cells are going to be genetically identical to each other and to the parent cell. So a cell does more than just divide. A cell undergoes an entire cycle as part of its life. So we're familiar with mitosis. We've spoken about mitosis before. Mitosis is when the nucleus divides, and it's the longest part of cell division. But cell division is only one part of the cell cycle. So I'm going to walk us through this diagram here. So we're familiar with cell division. We know that cells divide if an organism is growing. We know that cells divide if an organism has to repair damage, or if an organism, or simply if cells get too old and their life is over. So we know that cells divide, and we're pretty familiar with the process. Mitosis, which consists of prophase, metaphase, anaphase, and telophase, is when the nucleus divides. And then cytokinesis is when the cytoplasm, the rest of the organelles, the rest of the cell divides. So in cell division, we get... we're starting with, at the end of cell division, we now have two brand new cells. So the first thing that's going to happen to a cell after division is complete is the cell is going to grow. During this time, it's going to create some more organelles as needed, grow in size as it gains nutrients. And when the cell is finished this first growth phase, or G1 phase, then the cell is going to replicate its DNA. And we call this the synthesis, or S phase. Remember that in order for the parent cell to produce two daughter cells that are genetically identical to each other and to the parent, it's going to need to copy its DNA so that each daughter cell has a copy. So that happens during this DNA replication, or S phase. Right before a cell divides, it's going to go through a secondary growth phase, and that's just to ensure that the cell is big enough to divide again. So the two main components of the cell cycle are cell division, which is mitosis and cytokinesis, and then interphase, which is a growth phase, a DNA replication phase, and then another growth phase. And we can see from this, this diagram here that a cell is going to spend most of its time in interphase. So if we take a look at this table here, we can discuss a little, or we can go through a little summary of what's happening in each phase of the cell cycle. So interphase, I've referred to this before as a cell living its best cell life. It's doing whatever it needs to do to survive in order to keep, if it's our cell, in order to keep us alive, if it's a daisy cell, in order to keep the daisy alive. Most of a cell's life is spent in interphase. And as I mentioned earlier, there are three components or phases of interphase. G1, which is growth and normal function of the cell. S, or synthesis, when the DNA gets copied and again, the cell is functioning as normal. And G2, which is the second growth phase, is when the cell's preparing for division and again, just doing what the cell needs to do for part of its normal functioning. So mitosis, part of cell division, mitosis specifically is the division of genetic material, or DNA. Mitosis creates two identical daughter cells, and there's four phases of mitosis: prophase, metaphase, anaphase, and telophase. and we'll discuss what's happening in each of those phases shortly. Cytokinesis is also part of cell division. Remember that mitosis is when the nucleus divides. The cytokinesis is when the rest of the cell divides. And by rest of the cell, I mean the cytoplasm and the other organelles. Cytokinesis is the final phase of the cell cycle. When cytokinesis is done, we have officially 2 independent cells that are ready to enter interphase. Then the whole process can start all over again. So we are going to focus on animal cells here. And here we're going to just take a look at the a simple diagram of the cells in the various phases of the cell cycle. So here, interphase. Remember, interphase is not cell division. Interphase is when a cell is just carrying out its regular functions. So in interphase, we see a cell membrane, we see a nucleus, we see cytoplasm. And the important thing to note here is the DNA is in the form of chromatin. Now, chromatin is loose, uncoiled DNA. So remember, we've talked about DNA before. It exists in very long, thin strands. One strand of DNA is like 6 feet long. And so all that is coiled up in just one nucleus of one cell. So it's very, very thin, but very long. And so it's all loose in the nucleus during interphase. And when this DNA is loose and uncoiled, we call it chromatin. After interphase is when cell division begins. We're going to start with prophase. So during prophase, this is what we're going to see. We're going to see spindle fibers start to form. We're going to see the nuclear envelope or the nuclear membrane start to break down. And most importantly, the chromosomes, not chromatin, chromosomes become visible. So what that means is that long DNA, the chromatin, which is what we're seeing in interphase, has now condensed and to form the chromosomes. And remember that chromosomes form for the purpose of cell division because now the DNA is more organized. It will be and it can be divided much more simply in this form. So here we are in prophase, which is the first phase of mitosis. The chromatin has condensed into chromosomes. The chromosomes have something called centromeres in the middle. All right, so here's the second phase of mitosis. And the second phase of mitosis is called metaphase. So in metaphase, we see the spindle fibers lengthen and attach to the centromeres of the chromosomes. So the spindle fibers will tighten and the chromosomes will line up in the middle of the cell. After metaphase, we have anaphase. So in anaphase, the chromosomes will split. Each chromosome consists of 2 chromatids. Okay, so what that means is each side of a chromosome is a chromatid, and these 2 chromatids are genetically identical, which means they have the same DNA. So this is identical to this, and they're both identical to this. So in anaphase, we see the chromosomes split So each chromatid starts moving to opposite ends of the pole. The way this works is the spindle fibers will shorten, which is what pulls them apart. Finally, at the end of cell division, we have telophase and cytokinesis. Remember that telophase is the last phase of mitosis, and cytokinesis is the last part of cell division, so when the rest of the cell divides. So we're seeing both telophase and cytokinesis in this diagram. The chromosomes start to uncoil and turn back into chromatin. The cell membrane pinches, and this will ultimately separate the two nuclei into two separate cells. The nuclear membrane reforms, and there we have it. We have two brand new cells. And that is the end of our lesson on